part 3 of online lecture 3. We're almost finished with this topic of great experiments in molecular biology. We're just going to finish up with a few really classic experiments to really give you an idea of the wide array of experimental techniques that are used. One other thing that I also hope that you glean from this lecture is how science is distinguished from other disciplines, in that the conclusions that we draw are always based upon experimental data. What I want to do now is present to you the evidence behind the idea that DNA replication is bidirectional. Hopefully you remember from our chapter on DNA replication the discussion about this process and the fact that it occurs in both directions. We mentioned that there were multiple origins of replication and that the presence of those sped up DNA replication dramatically and were very important for eukaryotes, which have very large genomes. We also mentioned that bidirectional DNA replication occurred in prokaryotes as well. And of course what we mean by this is that DNA replication is occurring at both of these replication forks, which are indicated by the black arrows in this slide, but it's proceeding in two different directions. That replication then is happening twice as fast. So we presented that to you as a fact earlier in the semester. So what we want to look at now is, how do we really know that? What's the experimental evidence that showed us that DNA replication is bidirectional? And asking for the evidence behind a conclusion is the way all science works. The type of experiment that was used to generate the data that told us that DNA replication is bidirectional is a pulse chase analysis, or a pulse chase labeling experiment. And this is a method for examining a cellular process that occurs over time by successfully exposing the cells to some kind of labeled compound. This is called the pulse, and then to the same compound in an unlabeled form. And this is called the chase. And in most cases, radioactivity is used as a label. The reason for this is that we can detect radioactivity later on, particularly by using X-ray film. In terms of the bidirectionality of DNA replication, pulse chase labeling was used in a very famous set of experiments that were conducted in 1968 by Arthur Riggs and Joel Huberman. And they exposed the cells to a high amount of a labeled radioactive tracer. In this case, they used a radioactively labeled thymine, which they knew would be incorporated into the DNA, and then to a very low amount of thymine that had a very low label attached to it. And what they did was they actually zoomed in on a single strand of DNA right at where the origin of replication was, and they looked at where this high amount of radioactivity was incorporated. And so if you look at the slide that is shown here at the top of the screen, the arrow in the middle here is showing the origin of replication, and so they know that location by where the sequence is. And here's another origin of replication over here. And because of the nature of the pulse chase experiment, and they knew that they'd only exposed the cells to the high concentration of the radioactively labeled compound initially in a very short pulse at the beginning of the experiment, they knew that wherever the signal for the radioactivity was very strong, that had to happen very early in the process. So if you look at this drawing here, so here's the origin of replication, and then here's the concentration of the label. So this is on X-ray film. When you have a lot of radioactivity, it shows up as a darker black. And so here's your origin of replication, and you see that on both sides of that there is evidence of the compound that had high amounts of the radioactively labeled tracer. And then as you get further along, further away from the origin of replication, you start to see the tracer that was labeled in the lower amounts. And so what this indicates is that the events started here at the origin of replication. You have your darker label here and then the lighter label. So in terms of time, as you move away from the origin of replication, these are the first events, and then as you move out, those are the later events. The clue for why DNA replication was bidirectional here is that these darker bands are appearing on both sides of the origin of replication. That tells us that when the process started, so in its very few first seconds, the high label was incorporated and incorporated on both sides. And then as the experiment proceeded on, later events you can see this lower tracer being incorporated. And this was true when they looked at every single origin of replication. It's the symmetry on both sides of this origin of replication then that tell us that replication is proceeding in both directions. In our next example, we're going to look at telomerase. This slide is just a review to show you the action of telomerase. And again, when we were discussing DNA replication, we talked about the fact that normally the ends of the chromosomes in eukaryotes, which we call the telomeres, actually get shorter with each round of DNA replication, and that's caused by the removal of the primer on that lagging strand. And we talked about the fact that there were certain cells that actually continuously divide. And the way they get around this problem of telomere shortening was by the presence of an enzyme called telomerase. What I want to point out to you right now is that some of this work was actually just recently noted in the awarding of a Nobel Prize. And so in 2009, Elizabeth Blackburn, Carol Greider, and Jack Szostak received a prize for discovering both telomeres and telomerase. Now that doesn't mean the discovery was recent. They just actually won the prize recently. And so what they showed then was that part of what telomerase does is it serves as a template for the addition of DNA to the end of the chromosomes, but also that it had some enzymatic activity. And they showed that it was active in germline cells, so that's in cells that create sperm and eggs in humans, but also in some stem cells in eukaryotes. So how did they come about understanding the role of telomerase in the lengthening of the ends of the chromosomes? Or another way of thinking about that is in the prevention of the shortening of the ends of the chromosomes. So this slide is showing a couple of different kinds of data. So give me a few minutes to orient you to these slides, and then I will explain what we know about telomerase from them. First of all, you have two sets of data. On the right-hand side of your screen here, we have something that's called a southern blot. And a southern blot is just a way of visualizing DNA. In this case, in a southern blot, you can actually target just specific kinds of DNA that you want to visualize on the gel. And for these experiments, they wanted to visualize telomeres. And so what you're looking at on the right-hand side is, this is something that we call a gel, and it's a gel showing DNA fragments. And so you're going to run one of these later on this semester in lab, and I'm just going to try to explain to you very briefly how a DNA gel works. Basically, when the gel gets set up, at the very top of the gel, there's a series of what we call wells. And these are just places where you're going to spot your sample onto your gel. And in this case, what would have happened is they would have, everywhere you see at the top of the screen here where it looks like there's sort of a separate lane of fragments, that would have been one of the wells. Everywhere on this gel that you see something black, that actually shows you that there was DNA there and that it was some kind of telomere sequence. For this example, we're just going to focus on the samples that are at the bottom of the slide. And I'll talk in more detail about them in just a minute. So when I'm talking about the experiment, just keep in mind that we're focusing on the bottom part of the slide and that these black bands indicate the presence of a DNA sequence that's part of a telomere. The other thing you need to know about gels is after you spot your sample onto them, we typically take the gel and we immerse it in a buffer system, which is usually just a salty solution that will conduct a little bit of an electric current. And then we put a charge on the gel in such a way that there is a negative charge where the wells are and a positive charge at the bottom of the gel. And we do that because DNA actually has a slightly negative charge, and so it gets repelled by the negatively charged electrode and actually gets attracted to the positively charged electrode. So in your gel, you're gonna set it up in such a way that you've got negative charge near the wells and positive charge away from the wells. And then you actually just let this run for quite some time. And what I mean by run is you actually run an electric current through the gel. And what this does is it forces the DNA fragments away from the wells and towards the electrode that has the opposite charge. What we know about DNA fragments is that the speed at which they run is directly proportional to their size, so that the bigger fragments don't run very far, and the smaller fragments run a further distance. So as you're looking at this gel photo today, where the mouse is right here, right at the top where the wells are, those are going to be bigger pieces of DNA. And then as you move your eyes down towards the bottom of the screen, those are going to be smaller pieces of DNA. So even if you don't fully understand what I'm talking about in terms of using the electrodes and how we know that these are DNA sequences and why they're showing up black, what you need to really get to understand this discussion about telomeres is that big fragments are going to be closer to the top and smaller fragments are going to be closer to the bottom. So this is our southern blot. So what happened in this case is they took a DNA sample and they just ran it on the gel. And then they took some sequences, little bits of sequences, we call these probes, and they matched only the telomere sequences, and they actually had radioactivity, a radioactive label attached to them. And they took that probe and they washed it over this gel. And anywhere on this gel there was a DNA sequence that matches the probe, you then see this black signal when it's exposed to X-ray film. So I'm not going to ask you a lot of detail about understanding a southern blot, but for those of you who are curious, now you know all the details of this gel. Again, for our purposes right now, you just need to know small fragments are going to be near the bottom and large fragments will be up here on the top. Couple of other things and then we'll talk about telomeres. On the left-hand side of the screen here, WT, that stands for wild type. And this triangular shape you see at the top, this is indicating the number of generations that they let these cells grow. So on the left, where the part of the triangle is small, this is 0 generations, and then it progressed all the way up to 150 generations. So moving from left to right within one of those triangles, it's 0 to 150. All of these other sets of lanes with these italics, these three-letter codes that are in italics with a number, those all indicate some kind of mutation that made the telomerase be inactive. And then over here, on the left-hand side of the screen, this is showing you an agar plate that has these cells growing on them, and it's been divided into 4 in what we call quadrats, with the wild-type cells being plotted up here on the upper left-hand side. And this layout is actually duplicated on this plate that shows 50 generations, 100 generations, and also 150 generations. So what we have is in the upper left-hand column, we have wild-type cells, and then in the other 3 quadrants, we have the cells that are telomerase mutants, and those are the ones that are run on the gel over here. So let's take a look then at what's happening as the generations progress in the cells where the telomerase is active versus in the cells where telomerase is inactive. And so what we want to focus on are these black lines that are on the bottom of the screen down here. And if you look in the wild-type cells, you'll see that at the beginning of the experiment, 50 generations in, 100 generations in, 150, that these 4 blobs have, these 4 blobs of DNA have run the same distance on the gel, which indicates that in this case the DNA is not getting any shorter with each progressive generation, all the way up to 150 generations. So this is what happens when telomerase is active. Now let's look at our cell types in which the telomerase is mutated. And what you'll notice if you look at every single one of these, that the DNA fragments on the gel actually get shorter with each successive generation. So you can even see very easily that there is a set of DNA fragments and the size of them is much closer to the well when you get down to 0 generations. And then as the generations progress, the size of the fragment gets smaller. And how do we know that they're smaller? Well, they encountered less friction, so they ran further away from the well. And that's why I told you at the beginning of this experiment that that's the main thing you needed to understand about this kind of gel. Large fragments are going to be near the top, and smaller fragments are going to be near the bottom of this gel. The last set of experiments I want to talk about is some of the experimental evidence for how we know that crossing over or recombination occurs during meiosis, which again, this is something we just presented to you as fact. Some of the evidence for this was provided by Harriet Creighton and Barbara McClintock, and they actually looked under the microscope and looked at the chromosomes, and so they found what's called cytological evidence that recombination had occurred between the genes exchange between the homologs just by looking at the chromosomes and seeing that they were actually different. So if you look at this slide here and you look at the parental chromosomes and you see one set here in blue and the other set here in red, they have some somewhat different shapes. This one has sort of a squiggly end on it with another structure at the top, a cap that's on the top. And what they found when they looked at the gametes, what they would have expected from the parentals is shown here on the top. But when they actually looked in the gametes, they found combinations of chromosomes that could not have appeared unless material had actually been physically exchanged between the two. So that was the cytological evidence for recombination. There was other evidence as well, but that's beyond the scope of what we're going to talk about today. So this concludes online lecture 6.